Yeah, it's a pleasure to be here today amongst such wonderful tech makers, so thank you for having me. And yes, I'm going to introduce a little bit about my work, which is how I use R to advance our research in kidney disease. So a bit about me: I did my undergrad at Queen's in biochemistry. I then moved to the University of Oxford, where I did my PhD in interdisciplinary bioscience, and I'm now back at Queen's as a research fellow. And over this time, I've developed from no real coding experience in my undergrad and diving a little bit into bioinformatics to developing some skills in Python and pandas, and really taking a deep dive now in my postdoc using R to do some big data analysis. And I'm just pointing out a really good course that I did, which is free and online, R for Biologists, that I highly recommend to anyone who's interested in diving into this field. So why do we focus on kidney disease? Well, it's a leading cause of death. It was the 12th leading cause of death in 2017, and that's predicted to become the 5th by 2040. It can also result in end-stage kidney disease that requires dialysis and kidney transplant. So this has massive effects on patients and their families. The leading cause of kidney disease is diabetes, and with an ageing population, that's why we're seeing this increased prevalence of this disease. And the UN set out their goals, their Sustainable Development Goals in 2015, with Goal 3 highlighting that they want to decrease non-communicable disease by 1/3 by 2030. And scientists have highlighted that decreasing the global impact of kidney disease is a major part of reaching that target. So there's various methods to study kidney disease, and what we've learnt over the years is that we really have to use multiple pieces of information to piece together the true complexity of this disease. And at each parts of this jigsaw puzzle, we have to make sure the data is reliable and that we go through those quality control processes. We have variability that can be biological variability, experimental variability, or missing data. So we have to make sure the data is really cleaned and in a good way before we integrate it. And what's fantastic here in Northern Ireland is that we have the Belfast renal transplant cohort. For the last 50 years, they've been collating data in an array of the technologies that we see here. So that's looking at DNA modifications, gene expression. We can look at lifestyle factors, healthcare records. And what we do in the end is this annotation process where we can look at databases to find out if there's any interesting genes or proteins that come out of these analyses, what do they do and what's their function. So today I'm going to focus mainly on the genetic side and then touch a bit on the annotation side. So we know that there's genetic variation that can happen that can influence the onset and progression of kidney disease. And by identifying the genetic variants that influence this disease, we can improve our ability to diagnose the disease and also help treatment planning for patients for personalised medicine. But what's great is that when we identify these genetic variants that are involved in kidney disease, we can then see if there's any overlap with the genetic variants that we know are associated with other modifiable risk factors that might be coffee consumption or insomnia. In my instance, I've been looking into telomere shortening. Now, telomeres are these protective ends of our chromosomes, because each time our cells divide, our chromosomes lose a little bit each time at the very end. So we need to add this extra length to make sure that our coding regions stay safe for life. But we know that telomere shortening can be associated with premature aging, diabetes, cardiovascular disease, and what we want to see is, does this influence kidney disease? So what we can do is we can identify genetic variants that we know to be associated with telomere disease or telomere shortening, and then we can correlate these with how these variants are associated with kidney disease. And I've had a little bit of an extract here of some R code that we use to do this, and it's a technique called Mendelian Randomisation. And there's two libraries that I like to use, Mendelian Randomisation and 2SimpleMR. So there's different methods of these Mendelian randomization techniques, and you need to make sure that you pick the right one for your analysis and for your data set. Normally, I start with the inverse variance weighted model. It's very robust, and then you can carry out some other methods to make sure that your associations are still in place. They treat outliers differently. And also, each data point here is a genetic variant and how it's associated with either telomere shortening or kidney disease. And you'll see that there's error bars here, so that's how much these variants differ across the population or the cohort that you're looking at. And these variants that vary quite highly aren't weighted as strongly as these that aren't weighted, that are weighted more strongly. We didn't see an association in this analysis, but previous studies have. And what's great about two-sample MR is that it provides you the tools to harmonize your data and also to carry out multiple MR methods in one go. So I highly recommend using this tool. And what's great is there's an online version called MRBase, so you can kind of dive into some data sets that are already there, give it a go and see how you feel about the technique. And what's even more wonderful is that we might have our kidney disease data set, but we don't have a data set that we want to study telomere length in. So what we can do is harness studies that have been carried this out previously and pull that into our analysis. And there's a great online tool called the GWAS catalogue that you can look up for any sort of factor, such as coffee consumption, insomnia, as I said, and it lists all the genetic variants that have been significantly associated with that. And we can even pull that in to R using a library called MR Instruments. And by being able to share this data, we can save time, we can save effort, and we can save money. And what's even better is that we don't need to have access to their raw data with their participant information and all the sensitive information. We just need the summary statistics, so it helps with data protection as well. So once we have these genetic variants or these genetic modifications that we see in our chronic kidney disease data sets, we want to find out what do these genes or the proteins they encode actually do. And we can do that via annotation and gene ontology analysis. So we look at the molecular function, we look at the biological processes and the cellular compartments of these genes and proteins. And there's a really great R package that allows us to do this. And it also produces really lovely data visualization tools, which helps us with our interpretation. It's called ViSEAGO, and the authors have also created a really great tutorial online to help you every step of the way describe what you're doing, why you're doing it, and how to do it. So a little bit of a basic intro into the pipeline with some snippets of code from the tutorial that I just mentioned. So you start off with your genes of interest. This is in our case our kidney disease genes, and then you'll compare that with your background genes. So in our case it would be maybe the whole human genome or a subset of that. And you need to pick your background very carefully so that the comparisons you're making are accurate. Then what you do is you can search the gene ontology databases. There's a range of databases that you can use and you can change which ones that you pull in. And for a range of organisms from humans to fruit flies. And what you can do is see that the enriched gene ontologies that are in your genes of interest. And ViSEAGO is fantastic because it clusters the similar gene ontologies together. So this really streamlines our data, and it also produces a great heat map. And this can help us to highlight hits for future study. And that might be in cell culture experiments or in animal models. And this means that we can streamline those without wasting any money, and we can sort of have a more directed approach downstream. So this is an example of the heat map that we get out. I don't expect you to read all the info on there, but what's really great is it has the color-coded sort of heat map, shows you how significant the hits are. It's clustered your significant gene ontologies together, so you don't have to sort of read each individual line. You get an idea as you go. And I've highlighted one, which was Wnt signaling. So this heat map was generated looking at DNA modifications in those with kidney disease who had diabetes. And what we know is that the Wnt signaling pathway is vital for kidney development, but also it's disrupted in diabetes, especially the high glucose environment. So here, what we see is that our gene ontology analysis is reflected in the biological data that we have, but now we can look back and see what genes and what gene modifications resulted in that hit coming up. So that can help us to aid diagnosis or pull out hits for downstream therapeutic development. So to summarize, kidney disease is a leading cause of death globally, and we know that integrating multiple approach is really what we have to do nowadays to understand this complex disease, to aid diagnosis and treatment planning. There's genetic variants that are associated with the onset and progression of this disease, and we can use these not only to aid diagnosis, but via Mendelian randomisation, we can use it to also identify new causal risk exposures. And we can also do gene annotation to translate this knowledge into functional insights, which can be used in the future to aid diagnostics and therapeutic development. So I'll just finish with a very important slide to me is the acknowledgement slide. I wouldn't be here without all the people who helped me get here, and thanks to everyone for listening. There's some links and tools there. I'm happy for you to reach out and have a chat if you want to find out any more information, and I'm happy to take any questions.
Yep.
At what point do you see this, like research and development, being, able to use at scale, and, when will clinicians be able to avail of it?
Yeah, that's a very important question. So we're sort of at the before stages, so we're doing the sort of basic research that we're going to pick out these hits of interest. But there is a process later down the line that you have to validate those. So you have to look in datasets and make sure that it's going, if you're doing a diagnostic test, for example, you have to test its accuracy. So it is a long journey that we're kind of at the very beginning of, and it would be other scientists and researchers who would then take it on, or indeed also drug companies or diagnostic test companies. They would look at this fundamental research and then translate that into the clinic. So it could be 5 or 10 years at this point, but hopefully we'll get there.
Hi, thanks very much. That was really, really interesting. I was just wondering, do you see the development of these tools can also help in creating preventative, preventative measures and information to help people prevent these?
Yeah, you're absolutely
diseases?
right. Yeah, so we've looked, for example, at telomere length, but as I said, you can research the associations between all sorts of factors, particularly lifestyle factors, so coffee consumption, or there's a big study looking at sort of environmental factors, access to the green space, your activity levels. So if we could have these behavioural factors, and we could know more about how your behaviour translates into your risk of developing disease, then we could inform people about the healthy behaviours that they could take up as a preventative measure. And also, a key point here is as well, kidney disease, if we can diagnose it sooner, and also if we could categorise kidney disease patients into those that will progress faster versus those that might not progress as fast, we can also ensure that the patients in need get the right treatment. So we can also help prevent in that way as well.
Thank you. Any other questions? Cool. Thank you so much, Claire.
Thank you.